So the first question is, during PCR, which ion is mostly needed for the functional activity of DNA polymerase? The answer is magnesium. Sometimes manganese ions are also used, and they are both divalent or divalent ions, positive ions. So you will use salt as magnesium chloride or manganese chloride. Question number 2. A PCR machine can be compared to, so it could be compared to a thermostat. How many primers are used to accomplish PCR? The answer is two primers. Next question is, what kind of bonding helps the DNA to become double-stranded from complementary single strands? It is a hydrogen bond. Next question was, at 94 degrees Celsius, what happens to the DNA? The answer is denature. At 55 degrees Celsius, what happens to the DNA during PCRing? The answer is primer anneals. On the primer anneals. At 72 degrees Celsius, what happens to the during the PCRing? The answer is Taq polymerase begins to synthesize new DNA molecule, or it can be called as elongation. The next question is, during PCR, DNA is synthesized in which direction? So answer is 5' to 3'. Next question is, human DNA polymerase is not used during PCR is because it will get denatured at 95 degrees Celsius. In other words, the human proteins cannot stand that high degree of temperature. It's only the proteins from thermophilic bacteria can be used to do this. Resistant to this kind of temperature. In PCR, to amplify a target gene within a chromosome, we will use nucleotides as. So here the answer is, I repeat the question, in PCR, to amplify a target gene within chromosomal DNA, we will use nucleotides as DNTPs. During PCR, the primers should represent the target gene. Why a DID nucleotide is one of the major components of DNA sequencing reaction? The answer is chain termination during DNA sequencing is facilitated by dideoxynucleotide. Next question, how many primers are used during DNA sequencing? That's only one primer. Question number, next question, what will happen if we add all the dideoxynucleotides in a single tube? The answer is, if we put all the dideoxy A, T, G, C, the answer will be that there will not be any synthesis of DNA. Next question. A researcher is performing PCR to amplify a sample of DNA. Unfortunately, he forgot to add the DNA primer prior to starting the experiment. Which of the following result is he most likely to observe? So the answer is the reaction will be completely unsuccessful. Next question is, what type of instrument is generally used to complete a PCR reaction? Answer is thermocycler. Next question is, what are the 3 steps to PCR thermocycling? So denaturation, annealing, and extension or elongation. Next question is, after 4 cycles of thermocycling, how many copies of targeted region will be in the PCR product? So the answer would be 16. So I'm not sure how you calculated this question, but I can ask you similar question, but just change the number. So so let's say for the first cycle, this will be the number of DNA strands. During second cycle, it will be this. And similarly in the third cycle, this will be and so on and so forth. So let's say for the fourth cycle, this will be 2 times 2 times 2 times 2. That will be equivalent to 16. So that's the trick. They might give you 30 or something, or so, that's the trick to solve this question. Nucleotides used to stop DNA synthesis during DNA sequencing differ from normal nucleotide in which position? They differ in the third position. So in sequencing, you use dideoxynucleotide that doesn't have OH group at 3'. Next question, which of the following is needed in the sequencing reaction? So all of the above, DNTPs, dideoxynucleotide, primer, and all of these. Next, last question. What is the function of template during DNA sequencing? I repeat, what is the function of the template DNA during the DNA sequencing? So the template provides the sequence for which the sequence has to be determined. Any questions? Any question that you want to discuss? No question? So I mentioned in the last class, last class, that in next one of the classes I will be talking about the term paper. And what I have done is that I have generally, what happens that you get a term paper, okay, write about 21st century, or write about HIV, or write about rainy season. So here I have done a very structured term paper, and the reason is that I want you to have full knowledge of RNA or mRNA, right? So at least if you have very much clear concept of mRNA or RNA, you understand not only what is going on, but you can also predict, or if you understand about RNA, then you automatically understand about DNA, because RNA is derived from DNA, and as compared to RNA, DNA is just double-stranded. RNA is heterogeneous population. There are many kinds of RNA. RNA is much more complicated than DNA. And so as I mentioned to you, I want you all to be the expert in the field of, or at least keep up with this, um, with the knowledge of about RNA. It has grown a lot in last 5 years, and especially 2 3 years, especially this applied aspect of this. So, so that's what I would. So step by step, I'll get to the term paper, but I just want to, uh, so I am. What I intend to is to give you a very, like I want you to be, as I mentioned to you, I want you to be the expert, very knowledgeable. So there is nothing to, there's no suspense about it. You have to go and write, right? But I'd want to take this opportunity to teach you also in advance, is that you don't have to um, deal with you don't have to so you do your good job, and you're informed about it. So so I'll just just repeat, rather, if you have taken my 461 class before, there are a few slides from transcription and translation that I'm going to repeat, and that sort of becomes your basic concept. So that human body is supported by cells. Cells are supported by, just a second, I'm having some issue. Cells are supported by various organelles, and these organelles have their own individual functions. Right? They work in team, these organelles, but they are very independent of each other. Right? So, so cells, they form the fundamental building block. Identical cells, they form tissues, and identical tissues, they work together to form an organ. Organ then form a system, and then there are many systems that contribute to the successful running of our body. If you go in reverse direction, then body is supported by system. For example, nervous system. What's the job of nervous system is let's say memory, and pain, senses, reaction, emotion, and so this nervous system has become a central focus of research on neuropsychology, depression, anxiety, uh, many Alzheimer's, many things. So then this is supported by brain and nerves, right? Nerve tissue that can extend from, just like you have cables running, electrical cables running all across New York City, various streets, various homes. If there is a 20-floor building, then each floor, the cable runs running. So the way the cable runs, or electrical wire runs in the city, um, that's how the nerve tissue or neurons, they run. Um, so in brain is an organ that supports the nervous system, and brain has many kinds of tissue that could be glial cells. There can be cells that participate in memory. There are meninges that try to protect the brain. So there are many kinds of tissues that come together to form brain. And then each tissue is made up of unique cell. And a typical cell that helps in passing the electrical and chemical signal is called as neurons. And so why I took the neurons as neuronal cells, because as we age, they start to divide slowly, and they start to divide slowly. So any damage that happens to brain or nervous system, it's very difficult to put things back. If there is a cut in the skin, it could be a deep cut in our skin, skin will heal because the cells are continuously dividing. But in brain cells, the cells have stopped to divide or divide very slowly. So replacement or therapy is very, very challenging. So especially if if a person has Alzheimer's and other kinds of neuropsychological disease, so it becomes more challenging. So I'm using this concept to draw connection with stem cells that we'll study soon, maybe next week. But coming back to our body is, as contrast to heart cells, brain cells, cardiac cells, nephrons, which are kidney and other alveolar cells in the lungs, they divide very slowly. That's why, when COVID happened, when it infected lungs, a lot of people had hole in their lungs, and it was difficult to recover quickly, and people took months to recover if they're diagnosed at the right time. But there are cells which divides continuously for, let's say, 300 billion cells are formed every day. And these cells are example like blood cells, it could be skin cells, it could be hair follicle, it could be digestive lining, so and so forth. So division is fine, but division is supported by, as you see, mitosis, let's say meiosis. They need large amount of protein, spindle fibers, centrioles, cell division. So this protein expression has to be supported, right? So when a cell gets a trigger to divide, right, then only it gets starts to divide. It doesn't divide all the time. So, and that trigger puts the cell on the cell cycle, which is very regulated. But I don't want to go there. I just want to quickly come here that there are various kinds of genes, right? Not so people get confused between sometimes between chromosome and genes. Um, so gene typically is suggest, the name genes mainly suggest that sequence of DNA that expresses protein. And there can be structural DNA which supports the regulation of expression of gene that makes protein. So our chromosome is actually mosaic of genes of DNA that just stay there. And mosaic of DNA that express protein or do not express protein. And the gene that expresses protein, it could be RNA that could make a lot of proteins. For example, spindle fibers. For example, microtubules, cytoskeleton, histones, right? So these genes are expressed, highly expressed, multiple, lot of proteins. And there are some genes which make only one protein, right? Um, so there are various situations in the cell. So and so a DNA doesn't directly make proteins, but it goes through a transition step, that is RNA. There are many kinds of RNAs, but as far as protein expression is concerned, in a classical world, there are three kinds of RNA: mRNAs, messenger RNA, rRNA, ribosomal RNA, tRNAs as transfer RNA. mRNA codes for proteins. rRNA helps in protein synthesis. rRNA is part of ribosome. It also helps in formation of peptide bond. tRNA is transfer RNA that carries amino acid from cytosol to the surface of the ribosome depending upon the genetic code. And genetic code is triplet nucleotide that represents RNA. So this is how RNA polymerase. So this is the way, let's say if you're taking a New York subway and you're trying to hold on, there's a rush time, and you don't have a place to sit to, you hold on to that rod, either on the top or near your head, or on the sideways. You try to hold. The way your fingers hold that rod of subway, so that you don't get jerk, or so that's how, the same way, right, this RNA polymerase try to holds the DNA. So you can see here, on one hand, so let's say this is the thumb, forefinger, and this is, so that's how the DNA enters and from one end and comes out, and the new mRNA or rRNA or tRNA is synthesized. Now, also you should know that that there is a huge difference between the how transcription and translation happens in prokaryotes and eukaryotes, and then this is mainly because of presence of nuclear that compartmentalizes the cell cytosol. So within the nucleus, you have transcription, and this RNA is processed, and this is very important step in the design of COVID vaccine mRNA. This RNA processing step is very important. Capping, splicing is not needed because you're making it in a company, but capping and poly A, they are more important, and more importantly, capping. This is very important when this COVID RNA is made. And why I'm saying important, because when we are taking injection, or we are taking vaccine, or vaccine is being coming into our body through a syringe needle intramuscularly, forearm. So it is bound to give a reaction, immune reaction, because this mRNA is foreign. But there's the reason why scientists have minimized the immune response to that RNA. That RNA is basically belongs to COVID, is because by playing with the capping process, so I'm just highlighting, this is the process, RNA processing, capping. Capping determines our immune reaction to the mRNA that we are taking COVID vaccine, COVID RNA. And also, there are three types of RNA polymerases. I'm sorry. Polymerase 1, it synthesizes rRNA, ribosomal RNA. Polymerase 2 synthesizes protein coding genes. So there are two kinds of genes, right? One is protein coding genes, and there are genes which codes for mRNA. So let me just introduce to explain to you for clarity one thing. One is, you'll come across this term very often. What is gene expression? So of course, gene expression refers when a DNA is making RNA, and this RNA, let's say this is mRNA, and this is translating into protein, right? Now, even if you have, this is the first step, this is the second step. So this first step is also gene expression. This is also gene expression. So whenever DNA transcribes, it is a gene expression, it's gene activity. But it is possible that in one situation, it holds on till RNA. In the second situation, it goes on to protein, right? So look here, RNA polymerase 2 not only synthesizes protein coding genes, which is mRNA, they also synthesize miRNA, microRNA. MicroRNA is a small piece of RNA, around 22 nucleotides, and it helps in regulating gene expression, right? And RNA polymerase 3 synthesizes tRNA, 5S RNA, and small RNA. So I just wanted to review the 461 concept with you. And now I'm taking you to your term paper. Let me go to the blackboard. So here on the so if you go to the content area and you term paper guidelines. So I am teaching various classes, and in one of the classes I have to read maybe 2 term papers. In other class, another term paper, and so I have I just want to get everything on my desktop before the semester ends or before the final exam. So your deadline is May 10th by 11:59 p.m. May 10th. That's I believe Tuesday. So here I have posted uploaded some interesting articles. How mRNA medicine might change drug landscape. So treatment profile. Then basic knowledge about mRNA. Then I have a second series of, I gave you the basic concept, but I also have a little bit more about your term paper. As I mentioned to you that I want you to give at least basic information about term paper so that you get a starting point. And if I can, motivate you to do a good job, then, that will be very good. So I want to open slides. Yeah. So these slides which are on the black. So one thing I talked to you just now was about the basic concepts of from BIO471 slide. Now I'm talking about mRNA, and that is about that is more directly relevant to the term paper. Yeah. Something is happening. I'm just trying to work with this. Okay. So so let's say this is the DNA. This is a central dogma of biology. Right. So promoter do not code for any RNA. Promoter is upstream of DNA, where RNA polymerase binds. These are RNA coding genes, right? This is more specific. So DNA definitely codes for RNA. Out of this RNA, exons only code for protein, introns. So here RNA means introns plus exon. But when this RNA is processed, capping, poly A, splicing, then only the protein coding mRNA or RNA, goes to ribosome and the cytoplasm, and protein is synthesized or a polypeptide is synthesized. Now focus on promoter and terminator. Promoter is a launching pad, just with the way your airline or airline lands on the runway. That's how RNA polymerase try to sense what is the point where it has to land and then take off for transcription. Terminator is the point where everything stops, right? And these are non-coding, non-RNA coding or non-protein coding, whatever you could say. Now, this becomes interesting in eukaryotes, where you have an RNA coding genes could be a mosaic of introns and exons. So exons codes for amino acids, protein or polypeptide. And these exons, they come together, intron is removed, and introns are removed by spliceosome, right? So if there is any mutation at the boundary of exon and intron, then it can cause disease. RNA processing, introns are removed, right? So RNA processing could be capping here, 5' end cap, 3' end addition of poly A tail, and then removal of intron. So we are interested for in COVID vaccine or any mRNA derived vaccines, this capping becomes very important. This poly A tail becomes very important, right? Even some of the nucleotides become very important. Now let's zoom in more. In 5' and 3' end of eukaryotic mRNA, there is UTR. UTR stands for untranslated or untranscribed region or untranslated regions, and they don't code for protein. So this UTR is this untranslated region is both regions are on both 5' end and 3' end. And then there's a Shine-Dalgarno sequence that helps with the process of transcription and translation, mainly translation. And so this is prokaryotic mRNA, where there could be a string of complete mRNA for protein number 1, protein number 2, protein number 3. So these protein number 1, 2, and 3, they're independent proteins. There is no intron here. Now in eukaryotic mRNA, this capping becomes further complex. This capping mainly happens on guanosine nucleotide, right? And mainly by methylation. So you have to look where on which atom methylation occurs. Then this is UTR, untranslated region, 5', 3', and then poly A tail. This is the microRNA I spoke to you, short piece of single-strand mRNA. I'm sorry. MicroRNA is short, single-stranded 22 or 23 nucleotide long RNA. This, whenever mRNA molecule becomes double-stranded, then it could be degraded, which means that this mRNA will not make protein. So microRNA, it's regulatory RNA. They don't make protein, but they can target an mRNA, and then this RNA can be chopped off when it becomes double-stranded. So then this is these are various forms of capping that can happen. Cap0 can elicit maximum immunogenicity in our body, right? So we got mRNA, right? Many of us who took vaccine. So if we had cap0, then we would have more severe immune reaction. But scientists have developed various kinds of cap1, cap2, cap3, sorry, cap1, cap2, which is another M6A cap. So these types of caps, they minimize immune reaction to the vaccine. Otherwise, we will have more severe problem in mRNA vaccine. Right. So read this. Now, the second strategy to minimize immune reaction is to use pseudouridine. Pseudouridine is that nucleotide. So see here. Here, uracil is attached through nitrogen. Here it is attached through carbon. Look at the double bond here. It's changed. So pseudouridine synthetase changes this nucleotide. This is ribose, right? Hydroxyl on both second and third carbon atom. So this is, changed by, and this becomes pseudouridine. This pseudouridine is very special. This also minimizes the immune reaction. Otherwise, we'll have a lot of trouble. So this is some information about from mRNA point of view specifically. Then watch this YouTube video here. This is very interesting. How a lady who was an undergrad student, this is a story in the first part of the presentation, this lady, this scientist, who is right now, I believe, at a very big position in Moderna. So in right now, we are having vaccines from two sources. One is from Pfizer and other from Moderna. So I am just forgetting whether this lady is in Moderna at a very high. I think she's in Moderna, or that lady, before she goes into talking about mRNA or her research, she talks about how, as an undergraduate student, she got interested in research, and she found a mentor. And at that point of time, anybody, so her mentor was working on gene mRNA, and at that point of time, people were focusing on science. Also moves like fashion sometimes. So at one point of time, when she was undergraduate student, mostly things were about protein and DNA, but she started working on RNA. Very interesting story. Then she gets an opportunity to do PhD, and then when she does PhD, her funding collapses. Then she gets a position in US. She comes to, I think, Temple University, or someplace. I have watched this many times, but I am just not very 100% able to recall. Then here also she gets her various she gets her work gets disrupted by funding at various due to various reasons, and she keeps moving from one lab to the another lab, and somehow she is only working on RNA, and mainly her like at the end she finds a mentor who also believes like her that yes, mRNA can be a good vaccine or therapeutic candidate. And the reason is very, I mentioned in my earlier class that if you use proteins, to manufacture a protein at industrial scale to vaccinate the whole world, if we focused on protein till now, perhaps we would not have had vaccine for COVID-19. When you use DNA as a vaccine candidate, it is possible that DNA can integrate into our chromosome and can cause secondary problems, because there are cases when adenovirus was given and it got integrated into human DNA, and then it caused cancer. And also, I think it happened in Europe when some kids they got so mRNA. When you give mRNA, if you give DNA, then the DNA, if you have to inject DNA as a vaccine, the DNA has to cross two barriers: cell membrane and then nuclear membrane. When you give mRNA as vaccine candidate, it doesn't have to go to the nucleus, because it just has to go to the ribosome. So that was the hypothesis, that mRNA could be a better candidate for a vaccine. And the other point of argument, the resistance that they got, was that RNA is very, very fragile. It can be uh, cut into pieces very, very quickly. For example, you can leave the DNA, if you're doing experiments in your lab, you can leave the DNA on your bench for 10 years and come back after 10 years and leave your DNA. Nothing will happen to your DNA at room temperature, or even outside in the parking lot. But in RNA, it's a different story. It will get chopped off. It's very delicate, right? So read this, especially watch this YouTube video. You'll enjoy it, especially the journey of this lady. Let me just, but I don't know. Whenever I try to mm, show a YouTube video on Zoom, somehow it doesn't work. So you watch it. Then guidelines. So it's very simple guideline, right? Um, I think it's 10 to 15%. I'll check the syllabus, but I think it's 10%. It's your term paper is 7 to 10 pages. Try to be as detailed as possible, right? So that I can understand, right? Because most of you will write similar thing, right? So let's say if I give you some paper on HIV, it's possible that there are 20 students in this class. You will have different approach towards writing about HIV. Somebody will talk about vaccines, somebody will talk about epidemiology, Somebody will talk about clinical features. So you have that independence in that. Here it's more focused. You talk about RNA and address specific questions. So it's very simple guideline. It's 7 to 10 pages. There will be a summary that you have to provide based upon the question. Half a page, introduction, 1 to 2 page. Main content of the body, 6 to 7 page, conclusion, and future perspective, and the references that you're using to write this term paper. Simple. Now let's look at the questions. I hope I uploaded it. So yeah, so there are straightforward. So I have listed 20 questions. All of them are very straightforward. You just have to do research and write the correct answer. Detailed, planned, organized, and
Professor, I have a question.
Just a second, let me finish, then we can talk.
Okay.
I just wanted to show you last thing, then you can ask me a question. Yeah, the last thing I wanted to show you is that submission of term paper here. So you can go and you can upload your term paper from your end. Yes, question. May I know who was asking this question?
Rimsha?
Yeah, hi Rimsha.
Okay, so the questions you posted, we have to answer those questions separately, or we have to answer them in the term paper?
In the term paper. Those questions are your term paper.
Okay.
It's a very focused term paper. Yes, it's not like you write what you want. You write what I'm asking. Did I answer your question, Rimsha?
Yes, Professor. Thank you.
So take a read and this question. I have listened to that video. It's very interesting. I related to it because there's so many ups and downs in your career, especially when you're in science, you do research, PhD, and then you get inspired that how much focused she was. I think she started from, I believe, Eastern Europe somewhere. And at every stage, the person who helped her was her teacher, her mentor. So it's very important to have a mentor in your life. That mentor could be your teacher. That mentor could be your neighbor. You can have many, many mentors in your career, in your life for various reasons. I mean, COVID was, bad in a way because I used to also offer independent research, but somehow, because of COVID, then student perhaps are not aware that I also give a course, BIO472 Independent Research, where I sort of mentor students because it's not possible to have many students in your lab. So you get some students who are motivated to do a little bit of research, you work with them, you talk to them, but listen to the story of this lady and great work. Maybe tomorrow she's going to get Nobel Prize, but she's at a very high position now, I believe in Moderna, if I'm not wrong, or Pfizer, only one of these two companies. Questions?
Professor, I have a question.
Mm-hmm.
So the term paper isn't supposed to be like an essay format then. It's almost like, you know, how we do your homework, it's like that. Like we just answer the questions.
Yeah, but you have to provide a summary, right? You will develop your own interpretation, right? So let's say cook, right? Like you cook pasta or something, but you all will have your individual flavor to that dish that you're making, right? So you have to write your own summary, you have to write your own conclusion, and focused on these questions.
Okay, thank you.
Mm-hmm. In one way,
Hi, Professor.
yeah, go ahead.
Hi, Professor. I don't have any questions, but did you get me for attendance? Anika Thomas.
Yeah, Anika. Now I get it. Got you.
Okay.
Professor, I also want to ask if you got my attendance.
Who is
Kayla
that?
Bethiji.
Who?
Kayla?
Okay, Kayla, you got it.
Kayla A? Yeah.
Yeah,
Thank you.
you're welcome.
And did you get mine,
And me?
Professor?
Name?
Tatiana Ross?
Yes, I got you.
Okay.
Professor, did you get mine, Shamika
Professor?
Lawrence?
Yes, I got you, Shamika.
Did you get
Thank
Foster?
Yeah, Tia Foster, I got you.
Okay.
Professor, did you get me?
Name?
It's Nuffle Vajra.
Yeah, I think I got you. Yeah, I just got you now.
Ashita Basum is here.
Yeah, I got you. So it's going to be fun. And let's discuss in every class a little bit if you want to discuss a question. Any other questions?
One more, Professor. So for the term paper, so we're going to mention like we are answering this question number 1, or we're just going to simply write it?
No, you
without
write the
mentioning-
questions too. It will be helpful.
Oh, okay.
So I have made almost a Word template, Word document template. You just have to plug in your answers,
Okay.
and in agreement with that guidelines.
Okay. Thank you.
Welcome. Questions? And it's just not that we are discussing today. I'm here every day. I mean, like Tuesday and Thursdays, so you can talk to me anytime. But if there are no questions, may I end this class?
Yes. Thank you.
Okay. Have a good evening, all.
Good evening, Professor.